I hope that you already downloaded CellProfiler as shown on the slides, as well as some of the materials that you will need for today. You can access that either via the link here or with the QR code. And you can also download the slides from today over there as well. So, oh, what happened? Sorry, 1 second. Okay. So can everybody see my screen? Confirmations? Okay. So welcome to today's workshop, where we'll be giving an introduction to CellProfiler, particularly a beginner's guide to segmentation. We'll be following in parallel to the tutorial on our website regarding how to conduct beginner segmentation. My name is Kamal Hamid, and I will be co-presenting with Le Lieu, and we are both postdocs at the SIMNI group in the imaging platform at the Broad Institute. We'll also be assisted later on with the breakout rooms where we have the hands-on section with the TAs who are also postdocs in the same lab, Esteban, Paula, Shatavisha, and Suganya. So the outline for today's workshop is I'll start off by just a gentle introduction about quantitative image analysis, the what and the why, and then I will transition into the overview of the CellProfiler software, where then I will hand over to Le, who will continue to get into more detail about how the software works. And afterwards, we move on to breakout rooms where we have the hands-on exercise for building a beginner segmentation pipeline. And I'd also like to draw your attention to the QR code on the right, which is a survey that we would be very happy if you could complete, and it will help us to further improve our community outreach and how we conduct these workshops. So, as many of you probably know, image analysis has not always been quite so quantitative. What you're looking at over here is a beautiful image, or rather a drawing, of pigeon cerebellum drawn by Ramon Cajal back in 1899. This was before the technological explosion of cameras, computers, and so on. And so the only way that this was conceivably doable was to look through the microscope and then free draw the field of view that you're looking at. Of course, as you can imagine, there are a lot of problems with this because of it's tedious, it's difficult to reproduce, not quite quantitative, and error prone and biased, and so on. Thankfully, now in 2024, we have digital cameras and digital images. And here what you're seeing is a classical image that we see in a lot of image analysis software. But this image, when you look at it, actually, if you really try to zoom in, what you see is just an array of pixels. And at the end of the day, arrays of pixels are just a different representation of arrays of numbers. So if you try to look, for example, at what this image represents in a program like in Python, what you see is really just a list of numbers that correspond to different intensity values at the given location in that image. And so that's great. Now we have these images that are extremely quantitative because we have these arrays of numbers that can be, two-dimensional, like a two-dimensional matrix, which is what we're seeing here. There could be three-dimensional, four-dimensional, and so on in terms of channels, time, and so on. But the question is, how do we really make use of this quantitative data? How do we analyze it? Of course, we cannot rely on our human brains for this because human brains aren't quite built for quantification. We have evolved for survival. And that's why we evolved for pattern recognition, for filling in the gaps, for extrapolating. So, for example, if you look at this image over here, which is a rather famous image, your brain is likely tricking you into thinking that you're looking at a colored image. But then if you zoom in a little bit and you look at it, you see that it's actually a grayscale image with a superimposed colored grid that then fools your brain into allocating certain colors to certain objects in the image simply because of how our brains process visual input. And this extends beyond color. So even when we look, for example, at these grayscale images, I'm sure that a lot of you, your brains are tricking you into thinking that the nuclei of the two cells A and B have a different shade of gray, and probably also that the objects C and D are different sizes, where in reality they are the same brightness and the same size. So we are very prone to these kind of optical illusions, and this subjective nature of our visual processing means that we're not quite suitable for scientific image analysis and quantification. And for that, we need computational approaches. Now, a computational automatic image analysis approach typically involves a pipeline. This is what we term it. And an image analysis pipeline, in the most simple sense, can be divided into three segments. We have a pre-processing segment where you prepare your data, your images from their raw format into, for example, a more suitable file extension or format, a more suitable bit depth. You might have to align images, for example. And then you move on to the second component, which is segmentation. And this is what we will be talking about mostly today. And here you try to separate the different pixels in your image into classes of, for example, foreground, the background, different objects, and so on. And finally, you have the measurement section, which is where you now measure what you have segmented in terms of whatever parameters you're interested in, like intensity or texture or morphology, and so on. Now, the advantages of such automated image analysis pipelines is it extends beyond simply being faster and less tedious, where you can apply this pipeline to all your images at the same time, but it also quantifies heterogeneity at a single cell level. So, for example, if you look at these two images on the top left, what we see is that if you were to quantify the total stain of each color, you're probably going to see that there's a similar level of both of them. But if you try to look at the individual cell level, you see that the colocalization changes and you get different biological insight. So it's important to be able to look at a closer resolution of like the individual cells and segment these cells so that you can see any differences at their level. And of course, having an image analysis pipeline where you have segmented cells, for example, you can measure so many different properties at the same time, which is something not easy to do when you're doing it manually or subjectively. And for example, you can look at the cell shape and not just the stain distribution and so on. And of course, in the day and age where we have these enormous data sets, we can now distinguish subtle changes that we wouldn't be able to see just by looking at it by eye, simply because of the size of the data set, and also because some changes are simply too subtle for us to see. So now we have this great approach. We have the computational pipelines to analyze images. And the question is, well, how do we build these pipelines? We need certain tools to do that. And here enters the stage CellProfiler, which is a free and open source image analysis software. The development of this software started about 2 decades ago, started with Ann Carpenter, and ever since it has accrued quite a substantial number of citations, and it's been described as one of the most flexible and usable softwares in independent analysis. And but of course, CellProfiler is great, but it's not the only software out there for this sort of purpose. I'm sure a lot of you are aware of ImageJ, which evolved into ImageJ2 and then Fiji. And Fiji is a very versatile, powerful software that has the advantage of being very fast because it's Java-based. However, that usually only applies on the small scale in terms of speed. And it's extremely versatile because it has a huge number of plugins that have been contributed by the community and so on, enabling the measurement of a huge array of different types of images, including, for example, Western blots and agarose gels and so on. But it does have the disadvantage that it requires you to be able to write scripts or macros, whether in ImageJ's native macro language or in Jython, for example, in order to be able to apply an automated analysis from multiple images. And because of that, it's also often limited in automation because, for example, the language itself is meant to be simple and user-friendly, which limits its capabilities a little bit. And also because you have so many plugins, you sometimes have an issue trying to automate some of them or trying to make them compatible. On the other hand, we have, of course, another amazing software called Elastic, standing for the Interactive Learning and Segmentation Toolkit. And this is a machine learning-based software that uses, simple machine learning algorithm, random forests, in order to segment images. The advantages of the software is that it's very easy to use. It doesn't require any coding experience. And it can identify foreground and background in a large array of different types of images that might even be very heterogeneous, simply because you can train it in a sparse approach and it still produces a very robust segmentation. But the disadvantage of the software is that it informs primarily on object location, and it doesn't quite allow you to measure object properties. So you'd have to link it up with another software that you can then use the segmentation of this in order to measure your parameters afterwards. So CellProfiler tries to combine all the good things of these different softwares and tries to produce a very user-friendly, with basically virtually no coding experience at all actually required, and it allows you to build these pipelines for image analysis in your experiments. Now, when you open CellProfiler, you will be typically welcomed with this sort of window, which is actually itself rather helpful. What you can see here is that there are certain icons that you can click here in order to get different examples for you to try out the software when you're just getting started. You can go here to go to the tutorials, to see the list of tutorials that we have on the website, which extend well beyond the beginner segmentation, and of course includes PDFs and oftentimes video demonstrations. And you can also go to the Q&A forum that accompanies the software image.sc. And of course, this forum is not exclusive to CellProfiler and rather actually applies to a broad range of image analysis softwares, including Fiji and Elastic. And we encourage you to post your questions there for certain if you're stuck or stuff like that. And of course, CellProfiler is a very well documented software, so you can access the documentation over here, allowing you to get more information about how the software works under the hood. Now, once you go to the actual interface of CellProfiler, what you will see is something that looks like this. Now, in this interface, it can be divided into two panels. We have the pipeline panel on the left, and this is where your different modules are going to be placed, and these make up your image analysis pipeline. And on the right-hand side, we have the settings panel, which is where you can adjust the parameters and settings for the different modules that you select on the left-hand side. And a very important thing to note here is that there are these question mark icons, scattered across the software, and they're extremely useful because when you click them, you get a help pop-up window that corresponds to the whatever feature that is corresponds to, and it shows you the documentation for that feature, and oftentimes also gives you literature links if you want to do more reading on the matter from a conceptual standpoint. And on the left-hand side here, we also have the question mark that is dynamic, such that if you press or if you single click any of the modules, then you click the help button, then you get the documentation for that specific module. Now, to get a little bit into the pipeline panel, so the pipeline panel can be split into two types. We have input modules and we have processing modules. In the input modules, you have these four modules that allow you to essentially pre-process your images and decide how your naming convention is going to be and stuff like this. I will not be spending too much time on this because it can get slightly convoluted, and we do have a tutorial for that as well. And just to give you a little bit of a brief introduction about this, these four modules, you have the images one, which is a mandatory one, and that is where you drag and drop your images, and then you filter which, for example, types of files you'd like to accept into your image data set. And the metadata module, this is where you can provide the source of the metadata that you'd like to extract, if you'd like to extract from these images or from a separate file, like a CSV, for example. And in the names and types module, you have the specification of the types of images you're working with, like the dimensionality, the grayscale versus color, and so on. And of course, there's the groups module, which is typically has more niche use, and that is when you'd like to subset your images into different groups that will be independently processed. For example, if you're working with the plate experiment and so on. And an important thing to note here is that unlike other CellProfiler modules, these four are not linear and they work together in concert. And so if one of them is incorrect, then the other one will break regardless of the order that they are in. And as I mentioned, if you'd like to know more about this, there is a great blog post and a video tutorial on this that you can access here at CellProfiler input in broad.io. Now, to get into a little bit more detail about this, just in the input modules, when you'd like to start, the first thing that you'd like to do is to add your images into the software, and you do that by clicking the images input module and then dragging them into this field on the top over here. And the bottom field, this is where you select the extensions, for example, how you'd like to filter these files. You'd like to accept a certain extension or not. And you can also do a custom rule where you select the criteria for which files are to be accepted. And then you can apply the filters to the file list. And then what you will see is that the files get grayed out, the ones that are to be excluded, for example. In the metadata module, this is where you select whether you'd like to extract metadata and where you'd like to extract it from. You can either extract it from the file or folder names, and this is when you have a convention, for example, in the naming of your data set, or you can import it from a separate file, which would be a CSV file, or of course, you can also extract it from the image file headers. If you are to choose to extract it from the file names, then you have to use these regular expressions, which is basically an expression that dictates the convention of how you named your files in terms of channel number, plate number, and so on. And once you go through this, then you will see at the bottom over here how the metadata attached to the images looks like after you extracted them. In the names and types module, this is where you can assign user-defined names to a given channel, for example. So if you are to select, to establish a rule that images that come from a channel number one, for example, you can assign a name to this channel. And that is, in this case, for example, original text, which would correspond to your nuclear stain, for instance. And then you can also select the image type that you've extracted that follows these criteria, as well as how you'd like to extract the intensity range, whether you'd like to set it manually or from the metadata or simply based on the bit depth of the image that you're working with. And finally, this is the groups module where you can, as I mentioned, create subsets of your images for independent analyses or independent pipelines for them in case you're working with a data set that would require such a feature. Now, beyond the input modules, we have the processing modules, and this is where the magic happens. This is where the actual image processing occurs. And this is a dynamic pipeline that you build according to your needs, and you add or remove modules depending on whichever steps you have to go through in your analysis. And the important thing to note here is that at the bottom of this pipeline panel, you have an adjust modules segment, which allows you to add or remove or reorder modules. If you click the plus button, then you'll get a new pop-up window where you can search for new modules that you'd like to add, or you can browse the different modules that are categories that are there, for example, for image processing or for measurement, or you can even go for advanced tools for 3D analysis, for instance, or even go for Worm Toolbox, which is where if you have C. elegans data sets and so on. Now, another important thing to note here is that there is an important functionality in CellProfiler, and that is called the test mode. And this is very important, especially when you're starting to build a pipeline, of course, because in the test mode, this is where you can optimize your pipeline that you're actively building, such that you can run the modules on a single image set, for instance, instead of running it on all your images every time you'd like to tweak something in the parameters. And once you enter this test mode, you will see that there are a number of icons that will appear next to your modules. And what these icons tell you is basically what's going on with the different modules. So for example, if you have this green triangle, like the play button, this would be the next module that will be run once you click run. If you have grayed out this eye icon by clicking it, then what will happen is that whatever output comes out from that module will not actually be displayed, but it will still execute. If you click the pause button next to a module, then what will happen is that everything before that module will run, but this particular module will pause and everything afterward will not run. And on the other hand, if you have this X sign next to your module, that means there's an error somewhere in how you set it up, and then that means the pipeline will not run. Whereas on the other hand, you can also deselect a certain module by unchecking this checkbox, and this will mean that the module will not execute at all. But afterward, the modules afterwards will continue to execute. Whereas if you have this triangle that is telling you there's a warning that there's something potentially wrong with your module, but it will still run the pipeline. And in this particular case, the warning is simply that you cannot run export to spreadsheet when you're running in the test mode. Well, simply because you're in the test mode. Now, also when you look down at the bottom over here in this panel, you see that there are these 4 icons where the run button is, if you click it, then you run the pipeline until you hit the pause button. If you use the step button, it's exactly as it sounds. You basically go one step at a time. So one module at a time. And if you click the next image set, then what you do is you move on to the next image set, and then you start over your pipeline from the beginning. And this is particularly useful, for example, if you've just optimized your pipeline for a certain set of images, and then you're happy with it, and you want to test it and see how it goes for the next set, if it still applies the same parameters. And another very useful feature that is possibly a little bit hidden, which is this view workspace icon. And in this feature, if you click it, what will happen is you get a new pop-up window that will allow you to visualize some of the segmentations or processes that you've done. And you can select what you'd like to see. For example, you could select that you'd like to see a certain channel and how you'd like to color the outline of a certain segmentation. You could display certain measurements that you've made so far, and this would serve as a sort of sanity check for you to see whether what you're doing makes sense. And you could also use that, for example, in presentations if you're trying to show your workflow and the quality of your approach. Now, another feature that we have in CellProfiler is something called figure windows. And these windows will pop up whenever you have a module that has an output, for example, or if you double click your images back in the beginning when you imported them. And what the figure windows are simply just a way to display these images. And they, however, are dynamic and they do contain certain functionality. For example, in the toolbar menu at the top, you have the ability to zoom, you have the ability to pan, and also you have the ability to draw lines in order to measure things that are in the image. And this is particularly useful when you're in the process of optimizing your pipeline, and you'd like, for example, to have a threshold for size, and you want to get a rough idea, for example, of the size of the nucleus. And if you do that by drawing a line on the image, then at the bottom right, you get to see the length of this line that you drew. And on the other hand, there's also other information in this bottom toolbar, which is the intensity and the XY coordinates of wherever your mouse is hovering within the image. And if you right click on the image, then you also get a context menu where you can adjust the contrast or you can show the histogram of the image, which is, again, very useful for optimizing parameters such as the siding, which thresholding algorithm you want to use, depending on the shape of the distribution of the images, for instance. Now, with that, I'd like to hand over to Le, who will get into the good stuff, the image processing modules in more detail. And I hope that I was clear with my presentation.
Can you see my screen well? Okay. So yeah, I think you got the basic idea how the different module of CellProfiler works. So I'm going to continue to start how we can do the segmentation better. So we want to find out the objects of interest, and we follow the basic principle of classical image analysis segmentation. So that would be my image have my objects of interest bright and elsewhere else is dark. But in practical scenario, that's not always the true case. So for a better segmentation purpose, we basically can do some transformation that can be make our images with dim signal, but its true signal brighter, and I'll mask out the bright signal, but truly is, and they are not supposed to be segmented at the current time. And we have to think about that any transformation we can do to improve the segmentation quality is legal. However, I want to emphasize that we do this transformation for segmentation purpose, but in the downstream measurement, we should pay attention. We do measurement on the raw images instead of the transformed images. And the more transformation we made, we have to do critical sanity check. We didn't change the images too much, and we lose our attention of what the raw image should be, and we have and there are simple methods to check that, like we can do the outline over the raw images to say if your segmented objects is the truly objects you want to get. And we can do some image processing, and there are image processing modules in CellProfiler, and we usually use like enhance some edges or some enhance or suppress the features like speckles or smooth the images, or some advanced category. We can do the filter like median or Gaussian filters, and this not include all the CellProfiler modules that do image processing. And you can explore more for your own purpose. And then for the we design the CellProfiler pipeline, we usually follow two steps. First, we distinguish the foreground from the background, and then we how to split or merge objects that are touching together, and we can decide if they are one object or we separate them into two. In how we do separate the foreground and background, we usually use image thresholding method. So what it means is like you can see here is a histogram of the pixel, and in the lower end is the background, and in the higher pixel end is the real signal. And you can see in the if we choose the cutoff to say the left part is the background and the right part is the real signal. And if you choose two left, you can say that your segmented object contain a lot of signals from background. But if you go too far away from the background, you can get smaller object than you expect. So again, it's important to check if your method for segmentation is do the segment on the real objects. And usually we have multiple methods for the image thresholding. First is called minimum cross entropy. So it's good for like you already easy to identify foreground and background. But sometimes we, because for the maybe the stain is not even across the whole images, so there are several peaks in your whole images. So Auto is very robust for this case with multiple intensity peaks. And another method called robust background is ideal when your image has a lot of background and only some bright spots like Murphy experiments. You can choose robust background. And here is the CellProfiler interface. You can choose the different method for image thresholding. And what I introduced to you before, you don't need to memorize them. The beauty of the CellProfiler is it has a very great documentation, has the joint effort from the software developer and also the biologist. And also, like if you hook your mouse over here, the question marker, you can get the explanation of the different method is. So it's very handy. And also we can do some transformation before the thresholding in this interface as well. Because in some cases, like you can see here that you can see that there are some peaks in for the intensity histogram. However, you cannot really good to tell where to cut off from the background and foreground. So after the log transformation, you have a better idea like where to cut off them. After we got the primary objects, and then we can, in a lot of cell experiments, the cell are touching with each other. For example, these images, we can easily get that from our eyes that these are two separate objects. But how we can do that in the software? Basically, there are two methods. One is intensity-based, one is shape-based. So for the intensity-based method, it's ideal for the smooth objects. For example, like the bright spots. occur in the central part of the objects, and the boundary is more dimmer. And you can imagine here is a mountain that you pick up the peak of each mountain and separate by that. And the other is based on shape. Like if you can imagine, if two cells are roundish and if they touch together, there would be a indentations occur. So if your cells are more in the regular shapes, you can choose this method for your to separate different objects. And here again is the CellProfiler interface. You can use the method to distinguish the clump objects either by the shape based or the intensity based. And another handy module I want to introduce is the surprise local maxima in CellProfiler. What the local maxima means that the smallest distance allowed between object intensity peaks to be considered one object other than a clump. So basically, you can see here there are two local maximum intensity of two spots, and you can let's say if we measure the distance of these two different spots, let's say it's 5. And then if we choose the mean maximum distance equal to 3, and because it's less than 5, so they consider these two spots as two separate objects. But however, it's not the true case that we can tell they are one object. So we can increase the mean maximum distance to 6 because it's less than 5. So they consider as the one objects. So it's good for you to fine tune the different parameters to get the good segmentation. And here again is the CellProfiler interface to adjust these different parameters. However, that in the like ideally that for the segmentation, we want to segment each individual objects perfectly 100%, but that's not always the case in the practical scenario. So we can, it's good for us to start to fine tune the parameters. However, we can also know that where to stop. Like for example, for this experiments, you can tune the mean local maximum to separate these two. However, it can separate these two objects into two different objects. Or if you make it too larger, this one can merge into one per object. So a good theory would be that you check out the whole image and you get most of the segmentation good, and then you look at how much of the objects segment over segmented, or what the objects are under objects. So the ideal would be like they are over and under segmented there, but the ratios is around similar. And then you can move to the downstream objects. And in the biological experiments, we not only want the nuclei, we also want to get the information of the cells, the cytoplasm. So we can use the CellProfiler pipelines to find the primary objects. And you can see here is the output of how your segmentation works. And you can see here is your input images, and here is the nuclei, and different color represent different nuclei objects. And here is what we mentioned, that you can check out how your primary objects overlay on your input imaging. And you can see they have different colors, and different color represent different meanings. And green means that they are valid segmented objects for downstream analysis. And the yellow one usually means that they are invalid and because they touching the border. But of course, that you can choose if you want the objects touching the border or not. And we can also choose what is the cell diameter range in the beginning. And if they are outside of the range, they can label as pink, so they will be removed out from the object for the downstream analysis. And also in the output, you can get how many objects you can get from this segmentation pipeline. And as I mentioned that we want to find the nuclei, and we also want to get the information about the cell objects and the cytoplasm. So we also has the modules called identify secondary objects. But the logic for how CellProfiler pipeline design is that we assume there is one nuclei and we found it because it's easier to get the good staining of the nuclei, and also it's easy to get from the DIP channel. And it's based on assumption that one cell only has one nuclei. So if your samples has no nuclei or have double nuclei, this pipeline may not work well. So as we did previously, we already identify one nuclei, and then they will grow primary objects to get the cell. And if you have multiple channels, they can label the whole cell. Usually, if you have the whole cell stain, it will work better for this pipeline than a membrane stain. And for method to detect the secondary objects over the nuclei you already identified by the identify primary objects, there are two methods. One is non-image based, the other is based on image. And for the non-image based, so that's just based on the distance from the nuclei you identified, and it ignores totally about the image information. So for example, here is a nuclei you identified, so it grow the fixed distance to get your membrane information. So, and it has the donut shape. It's particularly useful when you don't have cell stain in your samples. Another method like image-based, and I want to highlight watershed image based method to do the cell segmentation. So for example, we have the different objects and they have they identify as different objects and they have the different peaks. So this method can help you you to connect this peak objects intensity and fill the hole around the different peak, and then you can get a whole objects. And then we can get tertiary objects by getting the nuclei and the cells. So it's very simple, like it's just the whole cells subtracted by the nuclei information. And so far we already get our objects for nuclei, cells, and cytoplasm. And in your own experiments, you may have the different markers. So based on this segmentation, we can identify where those different markers are expressed in the cells. In again, we can outline our segmentation of different compartments of the cells, like the nuclei or the cells. And we can look at the outline, or we can use RGB image with color-coded label. So it's very handy that you want to look at your image and identify if this particular area is identified as one object or two objects. And it's very critical. And if you do a lot for image pre-processing process, please do this steps. But it's very easy in the CellProfiler pipelines. And also, we can also detect the speckles within the nuclei, like you have fish experiments again, you have bright spots in your nuclei. You can also use this enhance or suppress features and mask images module in CellProfiler. And this part won't be covered by today's hands-on exercise, but it might be useful when you explore your own experiments. And again, after the objects and you are satisfied with your segmentation, we want to get measurements of your objects. So CellProfiler also has a very powerful measurement modules. It can measure the object size and shape, like area, parameter, or the length of the cells, or orientation, or solidity. And if you want to understand what these different shapes measures, you can go to the CellProfiler documentation pages to get the better understanding of what these different measurements are. Except the object size and shape, they also get the information of the intensity, and you can also get the mean, median, standard deviation intensities of the whole objects. And also, not only the intensity number you can also get the intensity distribution of the certain object. For example, if your object has the brighter spots in the central part of the object, or it has the brighter in the edge, so this measure object intensity distribution will tell you how this looks like. Also, you can also get the information of the different channel, like DIP channel and GFP channel, to see if there's two different signals are expressed in the same spots in the cell, and in other words, colocalization. And with this module, you can easily get the colocalization measurement, either the two different organelles or two different protein markers. And also, in some biological questions, we know that we care about the cells and the cell's neighbor. Like in one cell, how many other cells are neighbors? So in CellProfiler, they have measure object neighbors module. So you can get how many cells, like if here is your object, you can detect how many objects surrounding your objects of interest, and what's the distance of these this different neighborhood, and also the angle of the different neighborhoods. After all these steps, we finish the segmentation, we got the objects we want, we do the measurements, and we want to save all these things. So first, we can save the images with outlines or a lot of other options, and we can export our measurements in either CSV or TSV, and we can also have the option to export to database in a very large experiment size. And all this option and this export to database can be optionally make a property file for the CellProfiler analyst. And also, like, we fine tune the different parameters, different modules, and we want to save these parameters for our next experiments. So we have two options here. First, we can save in the projects. So it's save pipeline, image locations, and metadata in a file called .cp project. It's very good because everything will be saved in the same place, but it's always not portable. So there's another option. You can exporting the pipeline, only contain the pipeline in .cp pipe file. So it's more portable. And also, when you want to publish in your papers, it can be a supplemental material. And also you can select either saving projects or saving the pipeline, or choose the both of them by default in the preferences menu. I think here I want to conclude our presentation here, and it's just an overview of how the beginner of the using the CellProfiler, and you can find more information to suit your own experiments. And you can go to the CellProfiler website. There are a lot of videos, tutorials there, and a lot of documentation there as well for your own purpose and also for your exploration. And also, I contain the CellProfiler GitHub page here as well. If you want to download the CellProfiler from source, and because it's open source software, if you feel for some specific measurements or some specific thing you want to do but not include in the current CellProfiler software, you can write your own plugin in CellProfiler. And also, as Kamal present before that, we has a good open scientific community, and usually when you have something stuck for the image analysis, either in CellProfiler, Fiji, or whatever other softwares, you can post your questions there, or also search if someone else already post that questions and get the answer. So you can, it's very good to ask the questions there. And also, if you want to explore more advanced CellProfiler workshop, there are some more workshops related to CellProfiler as well. For example, you can use the connecting the CellProfiler to Elastic and CellPose. And also, when we get the measurements from CellProfiler, we always want to look at how what we can get thousands measurements and what this data can tell, because we are not a big fan of collecting the measurements. We want to get our scientific question answered. So the Morpheus is a software that can be used for the downstream morphological profiling analysis. And also, sometimes we want to include the deep learning based method for segmentation, and Piximi is a handy tool to start. So you may interested to have these 3 workshops in your calendar, and we can, if you have any questions, we can answer a few of them, and then we will send people to the breakout room, and you can do the hands-on exercise, and we will have TA assigned to the breakout room. And if you have any questions, you can also reach out to them. Okay. Do we have questions in the chat box?
We've been addressing the questions that were appearing, so I would say no. If anyone wants to make a question now, please feel free to write it either in the chat or in the Q&A box, or we can chat about it in the breakout rooms.
Okay, so I think we can.
So yeah, so
The
I will
question.
create a. Sorry, Sugani.
There's a question now, Lee, if you would like to address,
Okay.
or we can also then break out.
It's in chat.
Yeah, like the question is, can we also perform image analysis using RGB images, or should we perform on grayscale images?
I think the CellProfiler can handle either RGB images or grayscale images. It's just like you choose the image format in the input module. Okay, I think because the time we can send people to the breakout room, but feel free to ask any related questions in the breakout room. I'll send the slide in the Q&A section. Okay, so now we have
Okay, I will assign to the people now to the breakout rooms.
okay.
Okay, no worries. Yep.
There's also a follow-up question. How can we perform QC for images with CellProfiler?
Sorry, for me or Le?
No, I just noticed like there's another question in the chat if Le wants to take it as we join the breakout room. It says, how can we perform QC for images using CellProfiler?
Sheta, can you address that question?
Yeah, sure. Yeah. So thanks again, everyone, for joining. I hope it was a helpful exercise and you got to experience that profiler and also, learn something new. So before we wrap up for this session, quick reminder that there are quite a few other cell profiler related workshops coming up. So please take a look at the virtual I2K agenda. In case you are interested, there's one later today. There are ones tomorrow and the day after tomorrow as well. And final request to please fill out the survey that was linked in the first slide of the presentation material. It will really help the bioimage analysis community if we can have a better idea of what the current needs and challenges are. So yeah, with that, I again thank you for joining and hope you had a good time. And we'll hopefully see you again in one of the other tutorials. Bye.